everybody. Welcome to the last phase of cellular respiration. This is where we're going to talk about the electron transport chain. Like I said at the end of the last video, this is the super exciting part. We're going to do a lot of biological processes here, not just chemistry. But before we jump into that, I wanted to just review a little bit of the structure of a mitochondria. So back in chapter 4, you were learning about mitochondria, its general structure. So it is a membrane-bound organelle made up of phospholipid bilayers. So it's going to have all those same properties the plasma membrane does. It's selectively permeable, hydrophobic middle, all that kind of stuff. And then the inner membrane was wrinkled. All right, so very kind of basic drawing like we have here. So the inner membrane is also made up of a phospholipid bilayer, also has all of the same properties of the hydrophobic middle and all that kind of stuff. That wiggly nature of the inner membrane is important. It's the structure and function relationship. Imagine if we had a mitochondria where the inner membrane was just smooth. It didn't have any wrinkles in it. Well, when we start talking about this electron transport chain, this is made up of proteins kind of all set in a chain, like right next to each other, embedded in that inner membrane. So if you have, so I'll just draw a couple electron transport chain kind of sets here. So imagine if you had your inner membrane just smooth without the wrinkles. You could only fit so many sets of those electron transport chains. But when we go and we have wrinkles, we can fit a lot more of those sets of electron transport chains. It's increasing the surface, even though it's an inner surface, it's increasing the inner surface area of where those electron transport chains are. So over time, mitochondria that had the wrinkliest inner membranes were more efficient and were able to make more ATP. So that's how we probably went from maybe an inner membrane like this to what we see now as this wrinkled inner membrane, which we call the crista, are just the names of those wrinkles. And then inside, this is called the matrix, and we're going to be pumping some things and moving some things in these different areas. And this is called the inner membrane space. So the space between the inner and outer membrane is called the inner membrane space. So we have the matrix as a space, we have the inner membrane as a space, and we have the crista, which is that inner membrane that's all wrinkly, and that's where these electron transport chain proteins are embedded in that membrane. So they're going to be integral transmembrane proteins, and they're pumps. They're going to be an active transport pump that we'll be seeing powered by these electrons that we've gathered in our electron buckets. We're ready to dump them and let them do work for us here in the cell. So let's take a look at the basic idea of this whole electron transport chain, and then we'll take some details. So this is going to be a little animation. We'll get started here shortly, but each one of these colored boxes is representing one of those membrane proteins that will be embedded in the inner membrane. We'll see this on the next couple slides. But remember, electrons are being transferred from one place to the next, and those are our redox reactions. So here, if you remember, losing electrons is oxidation, gaining electrons is reduction. Now, in all of the first 3 stages of cellular respiration, we were oxidizing glucose and reducing our NAD pluses and FADs. So those electron carriers, our buckets, remember, they were being reduced, holding on to those electrons. Now we're going to turn the tables. Now we are going to take those electron carriers and we're going to oxidize those. We're going to lose those electrons and donate them to these proteins that are embedded in the plasma membrane, and they'll turn right back around and be oxidized and reduced, oxidized and reduced, oxidized and reduced as these electrons bounce from one to the next. There goes my cat walking across the desk. So those electrons, as they go from one carrier to the next and they gain electrons, they'll be reduced, and then they'll lose the electrons, they'll be oxidized, and they gain electrons, they'll be reduced, and they lose electrons, they'll be oxidized. And then we get to finally see where oxygen comes into play, because oxygen is going to be this final electron acceptor. So that's why we breathe oxygen, because it's got to play a part, because remember, it's really electronegative. It's going to be really grabby for those electrons. And so that's where they'll end up in oxygen as the molecule of water. So this is just showing you how the electrons move, but then also I want you to take a look at what's happening with this energy level. So this is our delta G. So I got to go back a little bit to that energetics stuff from chapter 7 that we we're talking about. If we have our products way down here, so let's say this is an energy of 5, and we start off as an energy of 50, just throwing some numbers out there, we do final minus initial is going to give us a negative 45. So a negative delta G is what kind of reaction? Because remember, this is an exergonic spontaneous reaction. So what that means is these electrons, as they're going from one carrier to the next, they are doing this spontaneously. It is an exergonic reaction, and we're going to harness that energy of this, each electron bouncing from one carrier to the next. We're going to harness that energy and allow it to do work for us in this electron transport chain. It's actually going to be driving active transport. So let's take a look at the more specific sets of these proteins that are embedded in the plasma membrane. All right, so here's our locations. I'll highlight these. We have the cytosol, so that's outside, just the cytoplasm inside of the cell. Here's our inner membrane space, and then here's the matrix, and here's the inner mitochondrial membrane. So I drew our little kind of stick figure mitochondria at the beginning of the lecture. All of those pieces are here, and then these purple blobs of proteins and enzymes, they are the sets of the electron transport chain that's embedded in that inner mitochondrial membrane. All right, so the first part of the electron transport chain is all of our buckets. Remember, our NADH and FADH2 buckets are holding onto those electrons that got stripped from the glucose and all of those intermediate carbon-containing molecules. So now, once we get to the electron carriers, here's all of those, and oops, you do not want a black highlight, that's not going to be good. Here are the NADH electron carriers. So let's do one of our redox reactions, because here we're like doing a reverse redox. So here's our NADH plus, I'll just say complex 1, goes to NAD plus, and I think the electrons go to complex 3 in this particular pathway. So where are our electrons? Where do they start and where do they go? Who's being oxidized and who's being reduced? So I know it's a little bit different than what we did before. It's kind of backwards. So if we kind of track on the NADH, it starts off as NADH, but ends up as NAD+. So is it gaining electrons to make NAD+, or is it losing electrons? So remember, electrons kind of like to hang out with the hydrogens. So if we have NADH with those electrons becoming NAD+, it's losing the electrons, and they're actually going to complex 1. Okay, so this is getting oxidized, and this is getting reduced, and these are our products. Okay, so that's going to be the case for all of those NADHs that we collected in steps 1, 2, and 3, glycolysis, acetyl-CoA step, and Krebs. We've collected all of those electron carriers, and now we're going to come and just drop off those electrons at the electron transport chain. Now for NADH, it drops it off at complex 1. FADH2 drops it off at complex 2. So that's just really the only difference, that they're enzymes. We can see our ASE endings, right? So each one of these is an enzyme, and so they have a specific active site. So complex 1 is called NADH ubiquinone oxidoreductase. That's a mouthful. Its active site is specific for NADH. For complex 2, it's called succinate ubiquinone reductase, and its active site is specific for FADH2. So just kind of our enzyme nature, kind of applying what we learned about enzymes and active sites and substrates. That's why they can't drop them off at the same place. Again, you do not need to know the names of these enzymes or complexes. You can, complex 1, 2, 3, 4, that's totally fine, or just electron carriers embedded in the membrane. All right, so here's our electrons. They're going to be bouncing from one place to the next, and eventually they're going to find their way to oxygen. And if you remember, our overall reaction was glucose plus oxygen gives you CO2 plus water, right? And we said glucose was being reduced to carbon dioxide, which we've done. We did that when we got rid of all of that carbon dioxide in the acetyl-CoA step and in the Krebs cycle. So we've completely reduced glucose by stripping, it's not, sorry, it's wrong. We have completely oxidized glucose into carbon dioxide. But where did those electrons go? So glucose had all those electrons, we oxidized it. They're being given onto oxygen because oxygen is being reduced to water. We haven't seen anything that has to do with oxygen so far. So oxygen was not involved in glycolysis. It was not involved in the acetyl-CoA step. It is not involved in Krebs cycle. And when it comes to the electron transport chain, it's like one of the very last steps that happens in the electron transport chain. And what happens are those electrons bop off that last complex, bind with a couple hydrogen ions and a half of an O2 molecule, which is basically an oxygen atom, and your end result is water. So this is the water that's produced by the reduction of oxygen, by the gaining of electrons with hydrogen ions gives us this final product. So by the time we are done with this particular part of the electron transport chain, we have completely oxidized glucose, we've used our oxygen, and we've produced carbon dioxide and water. That all the steps that we've done so far, we are done with our sugar molecule. But at this point, we've only made 4 ATPs. We had 2 net ATPs in glycolysis, and we had 2 ATPs per 2 spins of Krebs. So where does all this ATP come from? Well, there's one more piece, a complex 5, that's not shown here. It's going to show up on the next one, that actually is going to be the source of all of that ATP. But before we get there, I wanted to kind of show you what's going to happen, a little preview of what's going to happen with these electron carriers here. So kind of get rid of all this stuff. So do you remember in that last slide I had with the little animation of the electrons bopping down and they're losing energy? Well, one of the things that exergonic reaction is going to do, we're going to couple it, right? We talked about coupled reactions, how we can use an exergonic reaction to drive an endergonic reaction. So what if one of these protein blobs, these complexes, was an active transport pump, right? What if this inner membrane space was jam-packed full of hydrogen ions? If I want to move hydrogen ions against the concentration gradient, this is active transport, right? So I'm going to have to need energy to do that. When we were learning about active transport, we used the sodium-potassium pump as an example, and we said it burned ATP. It burned a molecule of ATP to pump something against its concentration gradient. That's great, that works. But here we have kind of free energy of these electrons as they're moving from one place to the next down this energy gradient. That is free energy. And these electron carriers use that dropping, that endergonic release of energy as the electrons are going down each one of these protein electron carriers, they're going to use that energy to pump protons. So what we end up getting is a very high electron gradient, so not electron, hydrogen ion or proton gradient. We have a whole bunch of protons hanging out in this inner membrane space. I love this because it's tying everything in. If you remember also back to chapter 5 when we were talking about, no, not chapter 5, chapter 7, when we were talking about exergonic reactions, diffusion is an example of an exergonic reaction. So when things like to go from high to low, that is a spontaneous reaction. Now hydrogen ions, because they're charged and polar, they cannot diffuse freely through this intermitochondrial membrane, this inner mitochondrial membrane. So they're kind of stuck in this little inner membrane space unless they can find a way back through a special protein carrier. And here we have one of those special protein carriers. This guy right here is called ATP synthase. It's the last part of this electron transport chain. So what it does is it takes that hydrogen ion gradient allows those hydrogens to go back down into the matrix following their concentration gradient. But because it's an exergonic reaction, we're going to use that to couple the endergonic process of phosphorylating an ADP into an ATP. I know it's a ton of words and language. That's what's so one of the benefits of doing these lectures this way is you can rewind. You can't rewind class if we're to be sitting in class. I can always re-explain things, but it's always nice to have this explanation, and you can go through it as many times as you need to. So let's kind of follow some electrons, and then we'll follow some hydrogens, and then we'll follow the ATP production. So here's our electrons, right? They're hanging out onto the ADP. They get released into complex 1. They travel, in this case, to complex 2, okay? In that process of these electrons being lost off of NADH, gained onto complex 1, they're losing some of their energy, and they're losing that energy, and that is going to pump these protons. So that's one active transport, right? So now we're in complex 3. These electrons are going to move to complex 4. And in complex 3, we're going to use that energy of changing their, going down their energy gradient. We're going to use that to pump protons. And then lastly, from complex 4, these electrons are going to go bind to oxygen to make water, and they're going to use that energy to pump protons. So we have 3 complexes that as these electrons are going down their energy gradient, they kind of start high and they go down, 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 like on that little animation. We're going to use that energy. We're harnessing that exergonic reaction to drive the endergonic reaction of active transport of hydrogen. And so we end up with this huge gradient of hydrogen ions in that inner membrane space. Okay, so that is our electron traveling. So we harness them all through our first 3 stages of cellular respiration. Now we're letting them loose to drive our hydrogen ion transport. So all of these hydrogen ions, pump, 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 we are basically building potential energy. So we have ions behind the membrane. It's almost like water behind a dam. So then here comes complex 5, this ATP synthase, which is allows hydrogen ions to go back down their gradient. So now this is a passive transport. This is a exergonic reaction right here, going back down because there's a whole bunch of hydrogens up here, very few hydrogens down here. So as the hydrogens are moving back down their gradient, that's an exergonic reaction driving the endergonic reaction of ATP synthase. And so that's this guy right here. To cram that 3rd phosphate group onto the ADP takes a lot of energy. And we get that by the hydrogen ions flowing back down through this ATP synthase. It's beautiful. And what's amazing about this enzyme is it actually spins like a turbine, just like a hydroelectric dam spins when water goes down it and generates electricity. This ATP synthase spins as the hydrogen ions go down it, it spins and mechanically shoves that 3rd phosphate group onto the ADP. And that's how we get our ATP product right here. There's a couple animations on the Helpful Links videos. I would encourage you to watch some of those animations that actually show the ATP synthase spinning. It's just so amazing. They, first of all, have been able to figure all of this out and then actually visualize it in live, like microscopic images. But I think what I have listed for you is just animations, but the animations are based on evidence and science that they've acquired in the lab. All right, so that is our electron transport chain. Very cool. It's the dropping off of all those electrons that we harvested in the first 3 steps, letting those electrons go down their energy gradient, being picked up by oxygen. In that process, we're pumping protons, and then we're letting the protons flow back down their gradient to make ATP. So we got a couple reaction couples here. We've got the coupling of the exergonic electrons driving the endergonic hydrogen pumping. Then we have the diffusion of hydrogen, which is an exergonic reaction, driving the endergonic reaction of ATP synthesis. And so this is constantly happening all of the time in all of your cells. You're making millions and millions of molecules of ATP every second in your body. It's just amazing. Okay, so this would be one of the animations. Again, I can't show you animations in this video, but you can go and take a look and click on those links yourself. I'm not looking for you to learn all the parts and pieces of this special protein, but it is kind of cool to see how the hydrogen ions start up here in their intermembrane space. They flow through this entrance port. It kind of spins around, and then they go out this exit port. And in the process of spinning, it drives this mechanical action of making the ATP. It's just really fascinating. Okay. All right, so here is our takeaway of what goes in, what goes out, and the why. So here are the things that go in. It's a really busy entrance, lots of reactants. And this I've made to per molecule of glucose. So for keeping track all the way from the very beginning, what are the things that we get as a payout of the electron transport chain here? So we get 10 NADHs. And you can see I've kind of broken those down as to where the NADHs come from. We get 2 FADHs from Krebs. Oxygen comes in from our breathing. Hydrogen ions are just part of the matrix in the mitochondria. And then ADP and inorganic phosphate are just part of the matrix. I just noticed my background. I don't have my lights turned on, and what time is it? It's like 8 o'clock. So as the lectures are progressing, my house is getting darker and darker. I can see the reflection is really strong on my glasses. Maybe I'll take those off. So into the electron transport chain, I didn't realize it's getting nighttime. All right, so those are the things that go in. So we've got a lot of reactants that are coming in. So lots of energy carriers. These were our electron buckets, right? So we've gathered all of those electrons in the processes of glycolysis, acetyl-CoA step, and Krebs. We get oxygen from breathing. Hydrogen ions and ADP are just part of the mitochondrial environment. What we get out is we get our water, because that's the oxygen binding with the electrons and the hydrogen. And here's our payoff. Wahoo! We get 32 to 34 molecules of ATP per molecule of glucose. Add that to the 4 that we got from glycolysis and Krebs, we have 36 to 38 molecules of ATP per glucose molecule. And the why? We just said it. We get a lot of ATP. We get a big payout for the food that we take in. This is what's going to help drive our cells. And then the last slide is just an accounting of all of those ATPs. Where do they come from? So, and how are they made? So we get 2 ATPs from glycolysis in the process of what's called substrate level phosphorylation, which is just the physical enzymatically driven adding of the phosphate group to the ADP. We get 2 ATPs from citric acid cycle in the same way, just an enzyme is hooking that 3rd phosphate group on to the ATP. So we get 4 via this process of what's called substrate level phosphorylation. Of the NADHs that are made in glycolysis, acetyl-CoA, step, and citric acid cycle, we get quite a bit. So we can add all of that up, plus the FADH2s that come from the citric acid cycle, and we get 32 to 34 ATPs made by this process called oxidative phosphorylation, which is just the redox reactions, driving the hydrogen ions, phosphorylating it via the ATP synthase. So oxidative phosphorylation is just a fancy word to describe the electron transport chain and how at the end of it we can phosphorylate the ADPs into ATPs. All right, that's it. So that's our electron transport chain. The coolest thing, right? It's cool. I'm a geek, but that's okay. You don't have to find it cool, but it's still important for you to learn this material. We have one more mini lesson I'm going to cover, which is kind of wrapping things up with cellular respiration and talking a little bit about fermentation, which is similar to the very beginning of cellular respiration, but it does not produce quite as much energy, and it's only like 1 step past glycolysis, so it's a little bit easier. Okay, so I will see you next time for the last video to wrap up Chapter 8. Bye.